Thank you, and thanks to the organizer for the invitation for USP to share our perspective on helping and setting standards for in this field. What I'll do, I'll recognizing that maybe this is probably the first time we give a presentation at ASGCT and not knowing really the level of understanding of USP. I'll spend the first few minutes on providing an overview on how compendial public standards evolved over time, and then talk about some of the standard setting activities in the cell gene therapy areas, and maybe take some time on a case study for how we developed a standard for to measure CD34 positive cells, and then share with you some of the upcoming work, the future direction that where we're going. So in terms of how we evolved, this is a quick snapshot from the first book that USP published in 1820, and fast forward 200 years later, we evolved from a book that provide recipes for how you develop, how you make pharmaceutical preparations, to a book that provides validated procedures to measure key quality attribute for pharmaceutical, biopharmaceutical product. The examples here are heparin and filgrastim. I think, but the concept apply to different type of therapeutic product where the core quality attribute, which is part of the CMC section in a regulatory filing, we'll look at some of the key tests for identification, purity, and potency or bioactivity, and those are like the examples that are provided here. The other evolution that I'd like maybe to highlight is as we evolved as an organization for setting standards for different variety of biotherapeutics, and you can look at from the top to the bottom, from what we think today is a simple molecule insulin, all the way to the bottom to as a therapeutic preparation based on stem cells with the CD34 marker. And I think maybe 80 years ago, insulin was not that simple. It was pretty complex. It was a complex mixture. But we did over the years is we developed standards which are in the form of monographs. Those monographs are the examples that I showed in the previous slide, where you have test procedures that measure key attribute and supported very often by reference standard that allow the user to make sure they pass the test. But as we worked on more complex product, and you can tell just by looking at monoclonal antibodies, which are much more better characterized than cell and gene therapy, we recognize there that given the complexity of the product, it is really challenging to develop standards that address a specific product. And from that moment, we started looking at standards that cut across and look at class of product or molecules like the monoclonal antibodies, and looking at attribute like variant and molecular variant, and also some of the impurities. And as we get to the CD34 stem cells, and I'll show you a couple of slides on that case study, it became clear actually that when we think of compendial standard, it's not about the product, it's more about performance of a method, performance of a process, and the type of standard that you can have to confirm that type of performance. This fits nicely within our strategy, which is really to develop standards based on really broad understanding of innovation and also regulatory development. And while we continue to develop standards and modernize those legacy biological product, the standards for those legacy biological product, and we took a shift to develop standards that cut across product and cover families or classes of product. We recognize that the cell gene therapy and other therapies are part of those emerging therapies where we haven't really done much work, but we need to look at standard in that area. And also part of our approach is really to go across the product life cycle. If you look at classic monograph that I showed, the one for insulin or filgrastim or heparin, we look at the product while it's on the market. So it's those key attributes to control the product while it's in the market. It's sort of like a QC type of test. But here in this area, we're looking across the product life cycle to make sure that we have standards that help support, demonstrate method and process performance, but also they can be broadly targeted at product families or classes. Why do we care about standard? Why do we need standards? I think this nice quote actually from a 2016 survey conducted by ARM, where the key point here is that product consistency and lack of standards is possibly the single greatest challenge facing the field. And reliable measurement for product characteristics to support informed decision making is really important during development manufacturing and to make it through the regulatory pathway. And a couple of areas where there's clearly a lack for harmonized standards, and in the cell and gene therapy field on the component and processes for product development. Lack of commonly accepted laboratory practices is something that has been highlighted, but also the need for quantitative validated and robust assay for the characterization of cell product. And lastly, the need for reference materials that allow actually the user to benchmark measurement and validation for critical assays. There are a couple of areas where we think we may be able to make impact, and that's where we're looking at raw materials, because at the end of the day, we think of the finished product, but if you look at a cell or gene therapy manufacturing, and in general, like for biological manufacturing, the number of raw materials used in a single manufacturing process is pretty high. I've seen actually processes that use up to anywhere between 50 and 100 type of raw materials. So you can imagine any lack of quality of one of the components actually can have implication on the finished product. So because of that, the raw material piece is really critical. And at USP, we use the term ancillary material to really refer to those raw materials that we use in the manufacturing process with the intent to remove at the end, but sometimes it's really hard actually to remove those materials. And I think it's important to pay close attention to the type of material that may end up in a product at the residual level. Analytical testing and methodologies is really another important piece. Cell counting and cell viability are two examples that are really critical when we deal with cell-based product, and the goal is really to improve consistency in measuring or just counting cells, but also to enhance method and processes for determining and interpreting cell viability. As far as product quality and characterization, cell characterizations is really important, and a lot of the flow cytometry actually based method are key here. Potency measurement and the need to have standardized method to measure product potency. And residual testing, as I mentioned earlier, it's really critical for some of the critical raw materials that are used in manufacturing processes. What we've done over the past maybe decade or maybe 15 years is address specifically some of the unmet needs in the cell and gene therapy. And it's not a lot actually, it's very preliminary at USP, but we tried to be foundational and have some of the critical best practices and standard approach for some of the materials and test procedures associated with cell and gene therapy manufacturing. And there is a series of chapters that are listed here, and I'll show you a snapshot from a couple of those where we talk about best practices, the type of quality testing that are required for cell and gene therapy product, but also special attention to the ancillary materials and some of the specific ancillary materials like bovine serum. We developed a standard over 10, 12 years ago, I think, and back then bovine serum was still used quite a bit actually in manufacturing. With a shift in moving away from serum to a cell culture free, to a serum free media, I think there's still interest in serum, but some of the early stages of manufacturing require serum, and we have some standard procedure that allow the user to qualify serum, but also calibrate against a standard that has been established through multi-lab collaborative testing. In addition to standards for some of the key growth factor and cytokines. So in addition to the documentary standard, the chapters, we do have reference standard, and I'll show you the case study for CD3 for positive cells. So in terms of cell and tissue-based product, and chapter 1046 is foundational in this area, and some of the key sections, and this is really like a table of content of the chapter with some of the key sections that address manufacturing best practices, some information on key component used in cell and tissue-based product manufacturing, the type of quality systems that are used in a facility that manufactures cell and gene therapy product. But also, I think one area where USP actually can contribute a lot is the consideration for validation of and qualification. And I think the validation and qualification covers both materials but also methods. So method validation, we do have a number of documents in USP that are not part of this presentation, but like 1225 is an important chapter in USP that is aligned and harmonized with an ICH document on validation of analytical procedures. What we did add over the years is additional validation document specifically to bioassay. So there is a suite of chapters, 1032, 1033, 1034, that cover the design, the validation, and analysis of biological assays. And the chapter 1046 will just make a reference to those, in addition to some of the general considerations for stability, storage and shipping, labeling, consideration for tech transfer, and regulation and standards. This is the kind of chapters that we included in the book 12 years ago, and it's been revised at least 3-4 times since then because of the development, the ongoing development in this field, the change in regulatory requirement, advances in technologies. So it's challenging for us, but it's something we're able to do with the support of stakeholders like all of you here, but also our expert volunteers who work with us on a 5-year cycle, and I'll come back to that toward the end of the presentation. The similar approach has been taken for gene therapy products. So again, not really talking about a specific type of gene therapy product, but some of the general best practices that apply to manufacturing of gene therapy product, some of the general consideration for vectors, whether it's viral or non-viral, but also additionally here some key information on the on-site preparation and administration. And again, one area where we spend, the chapter actually focuses a lot of attention on the type of analytical method that are used during development and production of gene therapy product, the type of approaches to validation, in addition to some of the general considerations for stability, storage and shipping, labeling, and some of the regulatory references to regulatory guidelines in this area. Another document that I want to spend a minute or two on is Chapter 1043, which was written initially to cover some of the approaches for qualification of raw and ancillary material used in cell, gene, and tissue engineered product. In reality, I think if you look at that chapter, it covers best practices for qualification of raw materials used in biological manufacturing in general. The chapter provides like a very nice overview of how you qualify a material that you introduce into a cell and gene therapy manufacturing. It talks a lot about the risk analysis and the risk, both risk classification of the type of material, a tiered approach like 4 tiers from a low to a high tier. With the lowest tier is really a pharmaceutical product approved for human use that you can introduce as a component of the manufacturing process, which sound like a safe and reliable material that you use in manufacturing, but for which you still need to do some level of qualification. But as you move higher in the risk to much more high risk material, the tier 4, where it's material that has not been developed to be used in manufacturing, that you introduce almost out of necessity because you don't have a choice. And there actually you could use it, but then the level of work and qualification is much higher. And that's what the chapter actually describes. It does actually talk a little bit on performance testing. So it's one thing when you have a material that you qualify from a safety standpoint. If a material has gone through regulatory approval as a pharmaceutical product and you introduce it in your manufacturing, so I think it has established safety profile, but you still need to demonstrate the suitability for use in that manufacturing process. So I think it's important to keep in mind suitability for use. And also about the residual testing and how actually it's important based on a risk-based approach when you do and when you don't maybe need to do residual testing for some of the material you use in the manufacturing process. So I think those are like the couple of examples that I thought are worth going through, and then I'll spend maybe 5 minutes on a case study for how we took a procedure that has been established to measure number of stem cells based on the CD34 marker, and how we made that procedure compendial and developed a reference material that support actually the measurement of this type of stem cells. So we're talking here about the enumeration of a rare event that requires specific gating instructions for detection. And the goal of this chapter is really to provide a procedure that a standardized single platform based on a flow cytometry method for the enumeration of CD34 positive cells. And the method can be used to quantitate CD34 positive in different type of samples: peripheral blood, leukapheresis product, bone marrow, and cord blood. This is, probably familiar with the iSearch protocol. So I think what we did, we worked with an expert panel, and Rob Sutherland was part of that expert panel. So I think 5-6 years ago, USP gathered this group, and we took that iSearch protocol and validated through a multi-lab collaborative study to make sure it can actually measure, number of stem cells in this different type of product, and enhance how that method could be used in a QC environment, or like in an environment where you need basically to have an accurate measurement of the cells. And as we developed the procedure into a USP document, we also worked in collaboration with the National Institute of Biological Standards in the UK on developing a reference standard. So we took a material that we developed into a system super reference standard that can help with assess reagent, but also calibration of instrument, and ensure like a correct gating for the data acquisition and analysis. And this is the procedure which some of you are probably familiar with, but it's basically a method that relies on the use of synthetic fluorescent microspheres, the beads, as internal enumeration count controls. And the homogeneous counting beads are added to the sample at a known concentration, and the wash steps are eliminated to avoid the loss of beads. And after the cells have been stained and processed, the counting of the beads and the cells are simultaneously analyzed on a flow cytometer, and the number of CD34 positive is calculated by comparing the absolute number of target CD34 positive cells and the number of counting beads detected in the same data file. The reference standard that we developed to go hand in hand with the procedure that is in the USP document 127 is derived from peripheral blood collected by apheresis from a GCSF mobilized donor, and it contains human leukocyte, erythrocyte, and CD34 positive cells that have been fixed and lyophilized. So it's a freeze-dried preparation of the cells. And the way it is used, I think it's just reconstituted in water and ready to use. No lysis step is required. And CD34, CD45 staining comparable to fresh GCSF mobilized has been established. Well-defined cell count and range assigned from a multi-laboratory collaborative study. So this is a study that an international study that we conducted with the National Institute of Biological Control and Standards in the UK with over like a dozen of labs. And the goal actually was to assess the reagent used to ensure the correct gating, data acquisition, and enumeration of CD34 positive cells. This is what you see to the right is the label of that reference standard. As we took this reference material for development through the collaborative study, using that multi-lab approach, the different labs tested vials of the candidate reference material using the method described in USP 127, which is based on that iSearch protocol. And the data was analyzed as a whole from the different labs, and it resulted on a label value of 1.24 times 10 to the 4 cell per vial. And after the constitution of that vial, so the user who will get this reference standard material, they reconstitute in 500 microliter of water and the mean concentration in 25 cells per microliter with a range of 16 to 34. So again, this is a system suitability standard, so I think we're not talking about an accurate measurement of what's in that vial. It's a range, and the way it is used to measure CD34 positive, like both for instrument calibration, for qualification of reagents, setting proper gating strategies, this is actually good enough for that type of standard. So I'm going to talk a little bit about how you can engage with USP and the opportunities for collaboration. And maybe before that, just a quick summary actually that speaks to, in this field, the complexity and variability of living cells, as well as the raw material used in the manufacturing, can create some challenges in characterization and production of the cell and gene therapies, and the importance of control of analytical methods, but also the quality of raw materials is essential to produce a consistent manufacturing. The control of procedures and analytical methods allow to reduce the non-biological sources of variability. There are different ways to get involved with USP. We do hold on a regular basis roundtables. We had one a few months ago on AAV vectors, and then back in the fall, one on mRNA-based therapies. And I think that one way maybe to get involved and advance actually the field of standardization of procedures and reference material to support this field. We do run collaborative studies. I just showed you an example of the collaborative study we run, and I think there's an opportunity to get involved with that. You can also contribute by joining a USP expert committee, and I have a page toward the end on how you can do that. There are a few areas that we prioritized, and I think we began to work on some of them. But the focus is obviously, as we highlighted earlier, is the raw and ancillary materials, and they're looking at the different type of starting material that are used across product, with few examples like media components, supplement, vector beads, also cryopreservation media and formulations. Some of the component of the gene editing based therapies like enzymes and guide RNA, and other areas for consideration in general like cellular immunotherapies, mesenchymal stem cells, and adeno-associated virus and lentivirus based therapies. The type of documentary standard and reference standard that we plan to develop or to mirror what we've already developed are like document that can cover flow cytometry standards for specific type of measurement, and also cell counting and potency assays, vector copy numbers and for viral vectors. Boro mentioned the vector copy numbers for lenti. That's one project that we initiated, and we're looking also at other areas like AAV. I have two of my colleagues here in the audience, Rebecca Potts and Jim Richardson, who can also share some of the information of the ongoing work. This is a diagram for how we run collaborative studies when we develop a reference standard. And you can tell, I think, at the front, actually, the early steps where we bring a bulk material, but also qualify a material to use it as a reference standard, and the type of characterization, but also as we go through fill finish and multi-lab studies. Those are the steps where we engage with collaborators. We have our own lab capabilities, but also engage with industry labs, academia, government labs, and contract labs. And this is the call for candidate, where we invite people to join for a cycle of 5 years to support the work that our expert committees do. We'll have an expert committee dedicated to cell and gene therapies in the cycle 2025. I'd like to thank you for your attention.
Thank you. So it is open for questions. Go to the microphone for questions, please. I see Fraser.
Oh, well, yeah, full questions. Thank you very much. Very informative. Just to try to just understanding ancillary materials, I guess I'm trying to think of in terms of viral vectors used for transduction ex vivo of cell products that go into humans, and also maybe going back in a different paradigm, the use of plasmids or recombinant viruses that go into the cell culture system to make a viral vector that then is a parental product. Would you define those? Are those ancillary products? And, you know, I think we've always had this debate, especially on the plasmid side, about how high a level of qualification or, you know, is required for plasmids, for example, in transfection for viral vectors.
So thank you. That's a great question. I think a part of the answer to that question will rely on regulatory expectation in a given region. I think there's a difference between US and Europe, for example, and I'm probably not the best person to dissect through that. But I think in general, if something is used, you know, upstream, and the level of qualification between something used upstream and downstream is different, because the more like a component get in contact with what is the finished product, the higher the risk. So I think that we're talking downstream. And the 1043 has those the tiered approach for qualification of raw materials, where it talks about even sometimes the need actually to rethink your strategy. I think if the ancillary material you're trying to use is a high risk, I think if there is no way that you can test, produce, procure an ancillary material of the expected quality, you may to rethink that. But there are other ways. It's resources also. It's how much do you want to add to a testing strategy for an ancillary material. So I'm not sure about the plasmid DNA. I think if anybody else knows from a regulatory expectation whether it's an ancillary or a starting material. I think in Europe it's referred to as a starting material, I believe. And I think that's maybe how much I can share. Yeah.
Thank you. Another question on the right.
Hi, can you please comment on potency tests for hematopoietic stem cells? What kind of tests and what kind of reference standard materials are you looking to develop for that?
The only one we looked at, and we really did not, we've been doing some work on paper, basically looking at what are the opportunities, is mesenchymal stem cells. And for hematopoietic stem cells, it was more looking at the type of assays, you know, moving away from a colony-based assay to other assays. And we haven't done studies to, so I think what we would like to do is engage with those who are in this field and see what type of studies we could run across using different type of methods before we set, you know, we put a document out with those methods. So it's really early stage.
All right, thank you.
Yeah.
So thank you all the speakers for the session and all of you who are attending the session, so we can close. So thank you, everybody.
Thank you.